cycloblastic with their epidermis and their gastrodermis. Gelatinous mesoglea, a gastrovascular cavity, the nerve cells organize into a nerve net, and their specialized cells we call as cnidocytes. This phylum would include 5 classes. The first one is class Hydrozoa, where cnidocytes present in the epidermis gametes produce epidermally and always released to the outside of the body. The mesoglea is largely acellular, medusae usually with a velum. Many polyps colonial, mostly marine with some freshwater species, including species of Hydra, Obelia, Gonianemus, and Physalia, which will later be shown. Then you have the class Kyphozoa, or the cap jellies. They are medusa in most of their life. Polyps are small. The gametes are gastrodermal in origin and released into the gastrovascular cavity. The cnidocytes present in the gastrodermis as well as epidermis. Medusa lacks a velum. Mesoglea with wandering mesenchyme cells of epidermal origin. They are mostly marine. An example of species is the Aurelia. Then we have the class Staurozoa, or the star-shaped jellies. They are medusa, absent, develop from benthic planula larvae, 8 tentacles surrounding the mouth, attachment to substrate by adhesive disc. They undergo sexual reproduction and are present in marine ecosystem. Example species would include Haliclistis. Then we have the class Cubozoa, or the box jellies. Medusa is prominent in its life history. Polyp is small. The gametes gastrodermal in origin. Medusa cuboidal in shape with tentacles that hang from other corners of the bell. They are mostly marine. Example species would include the Chironex. Lastly, we have the class Anthozoa. They are colonial or solitary polyps. Medusa is absent. Cnidocytes present in the gastrodermis. Cnidocyles are absent. Gametes are gastrodermal in origin. Gastrovascular cavity would be divided by mesenteries that bear nematocysts. Internal biradial or bilateral symmetry are present. The mesoglea with wandering mesenchyme cell has tentacles that are solid. They are mostly marine. Species would include anemones and corals. Species are Mitridgia. Here are some examples of our Cnidaria celenterata. First, the Obelia, the Hydra, and the Gonianemus under the class Hydrozoa. Next, we have under the class Kyphozoa, we have the Aurelia labiata. Then, we have under the class Taurozoa. As you can see, there are star shape at the end and even on its body, the Haliclistus sanjuanensis. Then, we have the box jelly, the class Cubozoa. Example is the Chironex flickeri. Then, we have the class Anthozoa, which would include Mitridium senile. Representative sea anemones, a sunburst or starburn anemone, or the Anthoplura sola. This anemone has specialized tentacles that it uses to aggressively defend its territory against genetically dissimilar anemones. This second one is a sea anemone, Dardanus calidus, living in mutualistic relationship with the hermit crab. They are these little anemones on top of the hermit crab. The hermit crab lacks a heavily armored exoskeleton over much of their bodies and seek refuge in empty snail shells. When this crab outgrows its present home, it will take its anemone with it to a new snail shell. This anemone riding on the shell of the hermit crab has an unusual degree of mobility. In turn, the anemone's nematocysts protect the crab from predators. This one shows the body wall of a Cnidarian, specifically a hydra. The Cnidarians are dipoblastic. They are having the epidermis and the gastrodermis. The epidermis is derived from embryologically formed ectoderm, and the gastroderm is derived embryologically from endoderm. Between these layers is the mesoglea, or the jelly-like formation in between the two tissue layers. Mesoglea is normally acellular in hydrozoa, but it contains wandering mesenchyme cells and members of other classes. In the class Hydrozoa, cnidocytes are present only in the epidermis. In members of other classes, they are present in both the epidermis and endodermis. Now, what we can take of is that these cnidocytes are the one that sting into our body when in contact. Now, let us learn about the cnidocyte structure and nematocyst discharge. Now, A. A nematocyst is one type of cnida or knights that develop in a capsule in the cnidocyte. The capsule is capped at its outer margin by an operculum, or the lid, that is displaced upon discharge of the nematocyst. The trigger-like cnidocyl is responsible for nematocyst discharge. A discharged nematocyst, when the cnidocyl is stimulated, a rapid osmotic influx of water causes the nematocyst to avert first near its base and then progressively along the tube from base to the tip. The tube revolves at enormous speeds as the nematocyst is discharged. A nematocyst armed with barbs and advancing the tip of the tube, it will be aided in its penetration to the prey as barbs spring forward from the interior of the tube and then flick backward along the outside of the tube, thus stinging its prey or its predator. Next is a figure of the generalized cnidarial life cycle. This one shows the alternation between medusa and the polyp body forms. The medusa right here and the polyp body forms. We will be seeing a transition from being a polyp going to a medusa. When we say a polyp, it is attached primarily to the soil of the ocean floor, while the medusa is an example of those jellyfish that we commonly see afloat on seas. The uiscious medusa produce gametes that may be shed into the water for fertilization. Early in their development, a ciliated planula larva will form. In this case, after a brief free-swimming existence, the planula settles to the substrate or the ocean floor and forms a polyp. Budding of the polyp produces additional polyps, this one, and medusa buds. Medusa will break free of the polyps and swim away. It will break away from the end point of its polyps. Budding of the polyp produces additional polyps and medusa buds. Now, the polyps or the medusa stage of many species is either lost or reduced, and the sexual and asexual stages, which would include a sperm, and an egg, will undergo sexual stages. While for asexual stages, it will be from planula to polyp and then going to medusa, which will later be incorporated into one body form. Now, to really understand the life cycle of a cnidarian, here is the abelian structure and life cycle and the Aurelian life history. For abelian structure and life history, it is an alternating between polyp and medusa stages. An entirely polyp colonies, this one, stands about 1 centimeter tall. A mature medusa is about 1 millimeter in diameter, and the planula is about 0.2 millimeter in length. Unlike abelia, the majority of colonial hydrozoans have medusa that remain attached to the parental colony, and they release gametes or larval stages through gonozoid. The medusa often degenerate and may be little more than gonadal specialization in the gonozoid. Next, you have the Aurelia life history. Aurelia is the uiscious, and as with a Scyphozoan, the medusa, which is approximately 10 cm, predominates in the organism's life history. The planula, which is 0.3 mm, develops into a polyp called as Scyphistoma, about 4 mm in length, which produces young medusa or ephyrae by budding. So, you can see right here that budding of the medusa coming from this point and then swimming away from the polyp, or